Around a decade or just over a decade ago, we began to see data emerging supporting the efficacy of a class of molecules called antibody-drug conjugates in breast cancer. And the first of these was trastuzumab emtansine, or TDM1. The idea being that there is a HER2-targeted antibody that is stably linked to a cytotoxic chemotherapy. The antibody then acts like a guided missile that goes to the tumor cell that's overexpressing HER2 and delivers the chemotherapy like a smart bomb directly to the tumor. This is a great sort of way of looking at it, and I think is exciting because one of the benefits might be that we could avoid the toxicity of chemotherapy and spare normal cells that don't have that antigen normally expressed. And indeed, the early data of TDM1 demonstrated women didn't lose their hair, they generally felt well on it, and it looked a lot better in terms of patient-reported outcomes and quality of life compared to chemotherapy with trastuzumab. These data led to a phase 3 clinical trial of TDM1 versus lapatinib capecitabine in patients whose disease had progressed after trastuzumab and taxane-based therapy in the metastatic setting. And indeed, TDM1 demonstrated an improved PFS as well as overall survival, leading to the FDA approval of this drug now about 7 years ago. So this has changed the standard way that we manage second-line breast cancer. TDM1 was looked at in the first-line setting compared to standard trastuzumab and taxane-based therapy and did not demonstrate an improved outcome. It looked fairly similar, but didn't demonstrate an improved outcome. And it didn't replace the THP regimen that had become standard since that trial was run. So our gold standard for frontline is THP, and second line is the antibody-drug conjugate TDM1. We haven't seen other ADCs come out in those 7 years since the approval of TDM1 that are as exciting until recently. TrustysMapDirecti, we began to see data regarding this agent. This too is a HER2-targeted monoclonal antibody, very similar to trastuzumab, stably linked to a very different cytotoxic payload. Instead of the payload hitting the microtubules, which is what TDM1 does, this payload is actually targeting topoisomerase 1. So it's a payload that we don't often use as a standard of care chemo for breast cancer. And the unique feature of this drug is the antibody-drug conjugate is comprised of a lot more of the cytotoxic payload than TDM1 was. So there are about 8 of the cytotoxic DXd payload per antibody with trastuzumab compared to 3 to 4 with TDM1. A phase 1b clinical trial was done looking at this drug, and in very, very heavily pretreated metastatic breast cancer, the objective response rate was well over 50%. So that led to a ton of excitement and, of course, development of larger clinical trial to confirm these data.